Hello, my name is Lara and today I'll be presenting astrocytosis as glial cell abnormalities. So first of all, I'll be discussing what glial cells are, the specific astrocytes, Hello, my name is Lara and today I'll be presenting glial cell abnormalities, specifically astrocytosis. I'll be discussing glial cells, astrocytes, and the pathology thereof, astrocytosis. So first of all, glial cells. Glial cells are very important in the central nervous system, and they have functions such as regulating the neuron's ionic environment, influencing the nerve signaling transmission speed, which is very important, managing the synaptic activity, and controlling the neuron transmission uptake, providing structural support for certain aspects of the neural development, as well as assisting or sometimes hindering the recovery of any injuries toward the central nervous system. There are different types of glial cells, such as the microglia, astrocytes, ependymal cells, satellite cells, oligodendrocytes, and Schwann cells. Today we'll be talking about astrocytes. So astrocytes are star-like appearance cells in the central nervous system, and they have a lot of functions that we'll be discussing today. So they maintain and support neurons. So the first thing that they do is they are very important in immunological signaling, regulating neural synaptogenesis. We have iron and water homeostasis, neuron transmitter recycling, as well as the blood-brain barrier creation and maintenance. So that's why it's so important for astrocytes to work optimally, so that we know that we can have all those functions performed well. So the first thing is the role in physiology. So the role in physiology has to do with the potassium and the sodium channels. They exhibit the regulated increases in the intracellular calcium concentration, which are crucial for the communication between astrocytes and neurons. Astrocytes contribute to the synaptic transmission through the calcium signaling and maintain fluid homeostasis in a healthy central nervous system through aquaporin 4 channels. Okay, astrocytes play a big role in maintaining iron homeostasis, and they generate chemical mediators that alter the blood flow, and with that they actually can regulate the cerebral circulation as well. Then the roles in synaptic activity and plasticity. Astrocytes regulate gliotransmitter release and maintain a balance between the excitatory and inhibitory transmission, which is crucial for brain health. They also play a critical role in neuronal circuit formation and maintenance thereof, facilitating cell-to-cell communication and recycling of neurotransmitters. They facilitate the development and growing synapses by releasing thrombospondin and release growth factors. Astrocytes are essential for maintaining the brain health by facilitating cell-to-cell communication and releasing of growth factors, which we just said. Okay, so the role in metabolism. So the basic thing that astrocytes do is have an effect on the hypoglycemia. So it indicates that astrocytic glycogen usage regulated by the transmitters can maintain a neuronal function during hypoglycemia and increased activity. The glucose metabolites pass through the gap junctions controlled by the neuronal activity and the transmitters, and as astrocytic glycogen degrades, lactate is produced and transported to the synapses and axons, providing energy during hypoglycemia. So this is very important in that state. Then the blood-brain barrier. So the blood-brain barrier is composed of two central units, which is our endothelial cells and our pericytes. So they line the cerebral microvessels and maintain a controlled microenvironment for the central nervous system. The tight junctions regulate the transport of essential molecules, including the energy metabolites, and is crucial for homeostatic site connecting the central nervous system and the system circulation. So it keeps the two apart but still in well function with each other. Then we have astrocytes that through their reactions with these cells support the dynamic nervous system controls and may produce the blood-brain barrier characteristics in cerebral and related epithelial cells. Okay, so let's start with what is astrocytosis. So astrocytosis in short is when there is an injury or an insult to the central nervous system, the astrocytes react wrong and they will actually respond in hypertrophy. So they will react in hypertrophy with their cells and their function. So there will be an excess of astrocytes in the central nervous system. So astrogliosis is a word used to describe the range of morphological and functional changes that occur in astrocytes following damage. The hallmarks of astrogliosis include hypertrophy of both the cell body, I don't know why that is blue, the cell body and the processes, as well as the overexpression of structural proteins such as vimentin or GFAP. Astrogliosis inhibits a very broad spectrum response. Furthermore, the more dormant astrocytes re-enter this cell type. Astrocytopathy and astrogliosis are two primary ways that astrocyte diseases typically present. I don't know what happened with this blue link. Any genetic, traumatic, toxic, or other injury can change an astrocyte's pre-existing role and capacity and refer to astrocytopathy. These illnesses impair the astrocyte network's capacity to perform one or more of its vital tasks. And then it can result in a range of neurological conditions such as Alzheimer's. So Alzheimer's disorder has the characteristics of apoptotic astrocytes. So numerous nerve system disorders include apoptosis, but one pathogenic characteristic of Alzheimer's disease patients' brains in the abundance of apoptotic cells. So in Alzheimer's disease patients' brains, during the autopsy, they actually found this abundance, this hypertrophy of apoptotic cells. Then we have the pathophysiology of Alzheimer's may include the astrocyte apoptosis. So the temporal lobe of the brain have a higher concentration of the DNA fragmentation positive astrocytes in a study done on the apoptotic phenotype of astrocytes in the brain of Alzheimer's disease patients. The brains of Alzheimer's disease patients shows an increase of terminal apoptotic astrocytes, and the density of this polymorphous protein deposits known as senile plaques, which is known as the main cause for Alzheimer's across the world, which can be either cord or uncored, was found to be linked with the density of apoptotic positive astrocytes. So when these astrocytes reach astrocytosis, they become hypertrophied and they become an excess in the central nervous system. They will actually become apoptotic and will not perform their function anymore. So as we just seen in Alzheimer's disease, we can find these apoptotic cells that are in abundance in the central nervous system, causing the brain to not function properly anymore. In conclusion, astrocytosis, also known as astrogliosis, is a disorder that causes hypertrophy of the astrocyte and its functions. It causes the astrocyte to lose its function in physiology, homeostasis, and the blood-brain barrier, and so on. The altered function of the astrocyte causes neurological deficits, specifically in that known as Alzheimer's disease. This is the references that I used, and thank you so much for your time.